Division of Vegetable Science, ICAR, New Delhi. Ma'am is going to deliver the lecture on genome editing technology for developing smart climate-resistant horticulture crops towards Viksit Bharat. Ma'am, we welcome you on behalf of the dignitaries, on behalf of NADCL, and on behalf of participants. Ma'am, you are audible. Please continue. The time allotted is 1 hour.
Okay,
Please keep
thank
five
you.
ten minutes at the end for the question answer session.
Okay. Thank you so much, ma'am.
Welcome, ma'am.
Okay, good morning, everyone. Myself, Dr. Sumal Lata. I'm working as a senior scientist in Division of Vegetable Science, and I'm working in two crops, okra and tomato, in the area of genomics and genome editing. And today I'm going to discuss about the genome editing technologies for developing smart, climate-resilient horticultural crops towards Viksit Bharat. So first of all, I'm going to discuss about the technologies, various genome editing technologies, and then their application part. In genome editing, we talk about the techniques which can produce genome modifications. The genome modifications like targeted mutagenesis, there are insertions, deletions, and substitutions. And we do these modifications at a specific site in the genome. So the whole genome editing relies on one principle, and that is the production of a double-stranded DNA break. That is created at a specific site, and after the creation of double-stranded break, it is repaired. So there are two repair mechanisms. One we call is NHEJ, and the other is HDR. So NHEJ is the non-homologous end joining repair mechanism, and this is error prone. And HDR is the error free, and it is the homology direct repair. So basically in genome editing, we create a site-specific double-stranded DNA break, and then this break is repaired. And for repairing, we have two mechanisms. One is NHEJ and the second is HDR. So to create the double-stranded DNA break, there are many kinds of endonucleases. So these are the endonucleases which are used for creating double-stranded breaks. One is meganucleases, uh, zinc finger nucleases, and the TALENs, which are the transcription activator-like effector nucleases. In the short form, they are TALENs. Then the fourth one is the CRISPR-Cas9, which are clustered regularly interspaced short palindromic repeats, CRISPR, and the enzyme is the Cas9, CRISPR-associated 9. And the fifth one is the CRISPR-CPF1. This is the another endonuclease from the Prevetola and Francisella 1, which is used for creating double-stranded breaks. So all these enzymes, they have been used for creating double-stranded breaks and for in the genome editing in various systems, in human beings, in plants, and in other organisms. So coming to the repair mechanisms, that there are two repair mechanisms. One is NHEJ. So in case of plants, the double-stranded breaks, DSB stands for the double-stranded breaks. They are mainly done using the NHEJ. And this repair mechanism, it operates in the cell cycle and it exhibits low fidelity in repair. Low fidelity means the accuracy is low. Means when the repair is being done, some incorrect nucleotides are also incorporated. So due to the error-prone nature of NHEJ repair mechanism or deletion of the nucleotides. So NHEJ leads to the sometimes complete loss of the gene functions, or insertion deletions, they are introduced in the exons, which leads to the missense or nonsense mutations. Now coming to the homology-directed repair, that is HDR. So in case of HDR, homologous sequence, it serves as a template to repair the double-stranded break. It allows accurate repair. The HDR is used to introduce precise nucleotide sequence modifications and gene replacement or insertions at target loci in presence of the exogenously supplied donor DNA as a repair template. And the HDR, it operates only during the S phase and G2 phase of the cell cycle, and it has lower efficiency, so it is more challenging in case of plants. So these are the two repair mechanisms, and these are the different nucleases. So this is the strand of the DNA. This black one represents the strand of the DNA. So we have these many endonucleases to cut the DNA, to cut both the strands of the DNA. Like we have the ZFNs, we have TALENs, we have CRISPR Cas9, and we have F1. So using these enzymes, this cut is made, and then after this cut is made, it is repaired in the cell. So for the repair, there are two mechanisms, NHEJ and HDR. So the NHEJ is error prone. So during the repair mechanism, sometimes deletions are created, or sometimes insertion is being made. So which leads to the gene knockout or disruption of DNA elements. While in case of HDR, we provide a donor DNA. This is the template we are providing. And then using this template, the repair mechanism is done. So this is the precise genome editing, and it is used for the gene replacement and gene knocking. So these two pathways operates, and these are the enzymes. In case of plants, mainly NHEJ is used because it has the higher efficiency. So coming to the different kind of the nucleases. So the first class of the nucleases which were used for genome editing, they are the meganucleases or homing nucleases. These nucleases, they are discovered in the genome of the microorganisms, and each nuclease has its recognition sequence. So they have the recognition sequence, suppose from here to here, the recognition sequence. So they have the recognition DNA sequence element from 12 to 40 base pairs, and they cut both the strands in a site-specific manner. So among the meganucleases, one enzyme CRE1 received the most attention, and they have been found effective in maize. But due to the rare occurrence of these sites, recognizable sites, it has the limitation that the sites are limited. Due to this rare occurrence of the recognition sites, the meganucleases, they have not been widely used by the plant scientists, but they are the first class of nucleases which were used for the genome editing. So after meganucleases, the first generation of the genome editing tool came, and that was ZFN, the zinc finger nuclease. So the zinc finger nuclease, they are generated by fusing a zinc finger domain with a fork1. So this is the enzyme, and these are the zinc finger array. So this zinc finger array, they binds to the DNA. They have the DNA binding domain, and there are several zinc fingers in different colors. You can see the zinc finger, this magenta color, then blue color, then this turquoise, and then this orange color. So these different zinc fingers, and each zinc finger recognize a 3 base pair nucleotide sequence. So you can see here that each zinc finger recognize 3 base pair DNA sequence. So in individual zinc finger nuclease, there are 3 to 6 zinc fingers. So this arrangement vary from 3 to 6, and they can recognize from 9 to 18 base pair long specific DNA sequence. Like in case of the meganucleus, this recognition size was 40 base pair, but here in case of zinc finger, it is 9 to 18 base pair. But zinc finger act in the form of dimer. There are two zinc finger required because the fork one acts only when it is in dimer form. So this is the arrangement of the zinc finger nuclease. These are the zinc finger array, two zinc fingers, and this is the fork one enzymes which cleaves the DNA at a specific site where the zinc finger binds. So so the specificity of this nuclease is the zinc finger array, and the nuclease is dimerized fork one, and they create the double-stranded breaks, that is sticky end is created. So the fork one nuclease must dimerize to cleave the double-stranded DNA. And to achieve the dimerization of the fork one, the two individual zinc finger nucleases, they need to bind to the forward strand and reverse strand. And the two target sequence, they must be separated by 5 to 7 base pair spacer sequence. So this is the spacer sequence, and these are the two zinc finger arrays. So the genome modifications have been done using the zinc finger nucleases in organisms like corn, tobacco, Arabidopsis, and soybean. But the challenge in case of zinc finger was the assembly of the zinc finger nucleases. They are technically challenging. And when we outsource zinc finger nucleases, it is expensive. So due to these limitations, these zinc finger nucleases, they have not been widely used by the scientific community. Then after zinc finger nucleases comes the TALENs. TALENs belongs to the second generation of the genome editing tool, and they were first used for genome editing in 2011. So, like zinc finger nucleases, TALENs, they are generated by using a transcription activator like effector DNA binding protein. So this is the DNA binding domain of the TALEN. These are the different tail repeats, and they are consist of different amino acids which binds to the specific nucleotide. So these tail effector proteins, they binds to the plant DNA through domain containing several amino acid repeats. 34 to 35 amino acids repeats are there, and you can see here in different colors that these amino acids, they binds to the different base pairs of the DNA. So in TALENs, the specificity is provided by the tail repeats. The nucleus is same, that is fork one, and the double-stranded breaks created here are the sticky ends are created. So this is about the TALENs. So the TALENs, they can be designed and they can be assembled to target any DNA sequence, but they are sensitive to methylation. If the that region of the DNA is methylated, we cannot use TALENs. And like ZFNs, two TALEN monomers, they are required to form a dimerized fork one. But compared to to ZFNs, TALENs, they are easy to assemble. Therefore, they had wider adoption as a editing method in plant, including the Arabidopsis, rice, brachypodium, barley, maize, tobacco, soybean, wheat, tomato, potato, and sugarcane. So compared to ZFNs, they have been widely used. But they are sensitive to methylation. So after TALENs, the CRISPR-Cas9 came, and the CRISPR-Cas9, it is regarded as the third generation genome editing tool, and this was used to edit plant genes in 2013, and it is the currently the prevalent genome editing tool. And CRISPR was discovered as a prokaryotic immune system that is present in bacteria that protect cells by selectively targeting and destroying foreign DNA such as viruses and plasmids. So when the bacteria they are infected by a virus, they are targeted using the CRISPR system. So the engineered CRISPR-Cas9 system is based on the type 2 CRISPR system, which has three components. One is the Cas9, that is CRISPR-associated protein 9, Cas9. There are two non-coding CRISPR RNA, the trans-activating CRISPR RNA and precursor CRISPR RNA. So this is the schematic representation of the CRISPR-Cas9. So the engineered CRISPR-Cas9, which is used in the laboratories, it has two components. One is the single guide RNA. So this you can see here in magenta color. From here to here, the 20 nucleotide long is the single guide RNA. And this oval shape is the Cas9. This bigger one is the Cas9, which is the endonuclease. Then it has this is guide RNA is also called as spacer sequence. So after this, there is a 79 nucleotide long region, which consists of the trans CRISPR RNA and CRISPR RNA sequence. So from here to here, this is 79 nucleotide, which consists of CRISPR RNA and trans-activating CRISPR RNA. So this spacer sequence is 20 nucleotide long, and it binds to the target sequence. So it will bind to the target sequence which we want to target, and it binds to the sequence containing a PAM motif. So that the PAM sequence for the Cas9 is NGG. So this is the PAM. So where the PAM is present, there only this spacer sequence will bind. So at the 5' end, 5' to 3' NGC motif is required to bind the spacer sequence. So it is present at the 3' end of the target. So this fused trans CRISPR RNA and CRISPR RNA, they form the stem loop-like structure. You can see here, this is the stem loop-like structure, and this binds to the Cas9 enzyme. And this Cas9 enzyme, it has two domains. One is RUBC domain and other is the HNH domain. So the HNH domain of the Cas9 is used to cut the DNA, which is complementary to the spacer sequence. So this is the spacer sequence. HNH domain will cut this strand of the DNA, and RUBC will cut the non-complementary strand of the target. So these two domains, they cut the strand, and they cut about 3 to 4 nucleotides above the PAM sequence, and they create the blunt ends about 3 to 4 nucleotides above the PAM sequence. So this is about the CRISPR-Cas9. So the specificity is provided here by the sgRNA, and the nuclease here is Cas9, and the break created here is the blunt end. In case of the talon and zinc finger, the sticky ends are created, but here the blunt end are created. So this is about the CRISPR-Cas, that how it was discovered. So this is a bacterial cell, and this is the Streptococcus bacterium, and these are different viruses which invades the bacteria. So when these viruses they invade, so different viruses are represented by different colors. So suppose this is the viral DNA, by the blue color. This green one is the viral DNA, and this orange one and this purple one. These are the different viral DNA which are incorporated into the bacteria when the virus infect the bacteria. So, and these black ones, these are the repeated sequences. So it constitute CRISPR DNA. Then this CRISPR DNA is copied, and it is used to make a CRISPR RNA. So this is a CRISPR RNA. So this is CRISPR RNA is transcribed. And so after that, this CRISPR RNA, this is the CRISPR RNA molecule. So the Cas9 binds to the CRISPR RNA, and this is the trans CRISPR RNA. So the trans CRISPR RNA will binds to the complementary sequence, and then the RNA all three comes and it cuts this RNA molecule, this CRISPR RNA molecule. So a genetic seizure is created, and suppose if this virus it infects again, then a bacteria will recognize it. Its DNA is recognized by the bacteria, and it is being cleaved. So this is the natural immune system which operates in bacteria against viruses, and this was used in the genome editing to engineer the plant and animal genomes. So suppose this is the guide RNA, this is the guide RNA which we want to target. This is the guide RNA. This is the trans-activating RNA. So this is the genome which we want to target. So this guide RNA will go and binds to this specific region, and then this break is created. And after this break is created, there are two mechanisms which operates, and then the mutations are created. So this discovery was done by these two scientists, Emmanuel Charpentier and Jennifer Doudna. And for this discovery, they received a Nobel Prize in Chemistry in 2020. So we have discussed that Cas9 is a DNA endonuclease which has two domains. One is HNH nuclease domain, and the second one is RUVC-like domain. And it is also involved in the CRISPR RNA maturation and CRISPR RNA guided DNA cleavage. Okay. And the trans-activating CRISPR RNA is the small trans RNA which has a sequence which is almost perfect complementarity to the repeats within the pre-CRISPR RNA, and which allow the formation of the RNA duplex, which is necessary for the maturation as well as CRISPR RNA guided DNA cleavage. And these repeats, they are the palindromes, and they are the short inverted repeats which can form the hairpin shaped secondary structures. So this we have discussed in the diagram. Then the CRISPR-Cas9 has been used in large number of species from rice, wheat, tobacco, Arabidopsis, in horticultural crops like tomato, potato, and apple, sweet orange, grape, lettuce, in watermelon. So it has been widely used, and the CRISPR-Cas9 is simple and easy to manipulate compared to the previously available methods like ZFN and TALENs. And it has high efficiency in producing the targeted mutations. And in case of CRISPR, the multiple scRNAs with different target sequence, they can be designed, and they can be used for the multiplexing also. When we are using more than one target, we call it multiplex. The technical simplicity and efficiency made CRISPR a true breakthrough in genome editing, and it allows even the non-specialized laboratories to use it routinely. Then there is another enzyme which is used, that is CPF1, and it is regarded as also regarded as a third generation genome editing tool, and it was adopted as a genome editing tool in 2015 and used for plant gene editing in 2016. Compared to the CRISPR-Cas9, it has two main components, that is CPF1 enzyme and the CRISPR RNA, which determines the specificity. Although these two systems, CRISPR-CPF1 and CRISPR-Cas9, they are similar, but there are some important differences. So this is the CPF1. So here you can see there are two components. One is the CRISPR RNA, that is 42 nucleotide long, and CPF1. So the CRISPR RNA, it has the spacer sequence that is 23 nucleotide long, followed by 19 nucleotide long repeat sequence, and this spacer binds to the target's DNA sequence, and this has the PAM motif, that is TTN at the 5' end, and this repeat sequence form a stem loop structure which binds to the CPF1. So this oval shaped structure is the enzyme CPF1. And CPF1 has two domains, RUVC domain and NUC domain. So the RUVC domain, it cuts the DNA strand 17 to 18 nucleotides away from the PAM sequence. And this NUVC domain, it cleaves the DNA at 20 to 23 nucleotides away from this PAM sequence. So it creates the sticky end, in contrast to the Cas9, which creates the blunt end, it creates sticky ends. So these are the differences between the Cas9 and CPF1 enzyme. So the CPF1 system doesn't need a trans-acting CRISPR RNA. It doesn't need this trans-acting CRISPR RNA, and which is required in case of Cas9 for CRISPR RNA maturity. And the CPF1 CRISPR RNA can efficiently cleave the target double-stranded DNA. So the engineered CRISPR CPF1 is about 42 to 44 nucleotide long, which includes the 19 nucleotide repeat sequence and 23 to 25 nucleotide spacer, compared to the 100 nucleotide scRNA in the Cas9 system. So these are the differences which we can see. First, in terms of the spacer sequence, here it is 20 nucleotide long, here it is 23. Then the PAM sequence is located on the 3' end, and it is NGG. Here it is located on the 5' end, and it is TTN. Then the domains, there are two domains here, and they cut the 3 to 4 nucleotides away from the PAM sequence. But here the domains, they cut 17 to 18 nucleotide and 22 to 23 nucleotides away from the PAM sequence. So therefore the sticky ends are created. So here we can see the differences. The specificity is provided by the single guide RNA, sgRNA. So the sgRNA is there. The nuclease is here the Cas9 and here CPF1, and the double-stranded break which is created is blunt end, and here it is sticky end. So in case of CPF1, it also functions as RNAs to process the pre-CRISPR RNA into mature single CRISPR RNAs. So in case of multiplexing, the CRISPR CPF1 system require only one promoter to derive an array of several small CRISPR RNAs, and it is even simpler tool than the CRISPR-Cas9, which needs to be separately express the individual sgRNAs when we attempt to edit multiple genes. So when someone is using Cas9 and using multiplexing, then he or she has to use different promoters. But in case of the CPF1, only one promoter is required for the multiplexing. In case of the Cas9, the blunt ends are created, and in case of the CPF1 system, the sticky ends are created. So, due to the creation of the sticky ends, the CPF1, it can produce the larger mutations compared to Cas9. In case of Cas9, the mutations are typically like one base pair, two base pair, indels, insertion deletion mutations. So, the sticky ends created due to the CPF1, they increase the efficiency of the HDR mediated donor DNA insertion in the CPF1 cleavage site. And there are differences in the PAM sequence. In case of Cas9, usually the G-rich PAM sequence is required at the 3' end of the target sequence, while in case of CPF1, a T-rich PAM sequence is required. So, the CPF1 system has been developed using these enzyme like Francisella novicida, Acidomycoccus, and Lectospiraceae bacteria. And the CPF1 system has been used for the genome editing in species like rice, Arabidopsis, tobacco, and soybean. This is the comparison of the different genome editing tools like ZFN, TALEN, Cas9, and CPF1. So if we talk about the target sequence, the selection of the target sequence, so the target sequence selection is limited in case of ZFN, and in case of TALEN, it is abundant, and in case of Cas9 and CPF1, it is very abundant. If we talk about the assembly, in case of ZFN and TALEN, it is laborious, and in the Cas9 and CPF1, it is simple. Cost-wise, the ZFN and TALENs, they are expensive, and these Cas9 and CPF1, they are cheap. Then the multiplex editing is difficult in case of the ZFN and TALEN, and it is easy and very easy in case of Cas9 and CPF1. The efficiency is low and moderate in case of the ZFN and TALENs, and it is high in case of the Cas9 and CPF1. Off-target effects. Off-target effects are low in all the system. Off-targets are the regions which are other than our target site. The genomic regions which we do not want to target, but sometimes the guide RNA binds to those regions. So the off-target effects are low in all the systems. And if we talk about the overall evaluation, so it is good for ZFN, very good for TALEN, and these two systems, they are excellent. So this is another comparison of the genome editing systems. So this we have talked that the CRISPR-Cas9 and CRISPR-CPF1, they rely on a single RNA molecule, that is the guide RNA or CRISPR RNA, to direct Cas9 on CPF1 to its complementary DNA sequence. They need the presence of the PAM sequences, and the analysis of the 8 plant species showed that there are 5 to 12 NGZ PAMs can be found in every 100 base pair of the DNA sequence, and 3.5 times more TTN PAM sites then the NGZ PAM sites can be identified in Arabidopsis. So in 100 base pair of DNA sequence, you can find 5 to 12 PAM sites of the Cas9, and more than this, sites of the CPF1. So if we talk about the practical part, and that if someone wants to do the editing, so what are the steps involved? So the first step is the synthesis of the guide RNA, or designing of the specific gRNA spacers. And for this, there are many web tools are available, like CRISPR plant, CRISPR P, Chop Chop. So these are the tools which are widely available and freely accessible. So one can insert the gene which one wants to target, the gene sequence or the gene ID can be given, and then the gRNA, they are synthesized by the software. Then we can screen, there are many gRNAs they are provided, so we can select and use a few of them for our experiment. So so if we talk about the applications, so the applications are that the most important application is the single gene knockout, that we can target the genes and we can create the knockouts. And the NHEJ is the main pathway which is used to repair the double-stranded breaks in case of plant. So this process can introduce the deletions and insertions, which are usually smaller than 100 base pairs. And due to creation of these indels, the frame shift mutations they are created, which results in the loss of the gene functions. And the mutations created by this CRISPR-Cas9 or CRISPR-CPF1 or any of the genome editing tool, they are stable. The mutations are stable and they are heritable in the future generations. So, so the due to the simplicity and the high efficiency, the CRISPR-Cas9 is the dominant tool for knocking out the genes. Then we can do the multiplex gene knockout also, that more than one genes can be targeted. And so the multiplex gene editing is not only useful for the functional genomics, but it also used for the study of the redundant gene families. Redundancy means the gene families which are having the similar kind of functions and the functionally related genes. And it allows the multiplexing, allow the fast bioimaging of the multiple traits. Then we can create the large fragment deletions using the CRISPR techniques. So how these deletions are created? Suppose when two double-stranded breaks they are introduced on the same chromosome at a certain distance. Suppose we create two breaks on the same chromosome at a certain distance, then there is a possibility that these two sites may connect through the NHEJ, resulting in the deletion of the intervening sequence. So, so sometimes these large deletions they are created, and the large deletions they are useful for the basic research for the study of the gene clusters or non-coding RNAs. So the TALENs and CRISPR, they have been used to produce large deletions in the species like rice, Arabidopsis, and tobacco. Then in case of the polyploid crops, where the knockout mutants which are difficult to obtain using the traditional genetic approaches, this genome editing tools can be incorporated to study the gene functions. So, so the genome editing technology they have been applied for gene knockout in triploids like citrus and apple, tetraploids like cotton, pasta, wheat, and potato, and hexaploids like camelina and red wheat, and octaploids like sugarcane. So in case of polyploids also, the genome editing system can be employed. Uh, then gene targeting. So the gene targeting is the use of the genetic engineering techniques to produce one-to-one substitution of a DNA fragment, that you are replacing that fragment, or you are inserting a new sequence in a specific genome loci, that is gene knock-in. So the gene targeting has many applications, like in the precise gene modifications or epitope tagging of the endogenous proteins. So in case of the agriculture, it can be used for the point mutations or indels at specific loci in the coding region or the promoter region. So making gene targeting also useful for the crop improvement. So then the precise base editing. So in many crops, the important agronomic traits, they are determined by the variation in one or few bases in the genomic sequence. Sometimes a single SNP is responsible for a favorable trait. So that desired sequence can be introduced by the HTR mediated gene knock-in. So an alternative to the gene knock-in is the base editor technology. This CRISPR-Cas9 base editor technology recently developed, which allows the editing of single base C to T. So single base can be edited, which results in C to T or GA substitutions. So without double-stranded breaks or exogenous donor DNA, single base can be edited. So in base editing, the sgRNA is used for the site where we want to edit, and here we use a modified Cas9. So what is modified in this Cas9? The endonuclease function is replaced with the cytidine deaminase, which can convert a cytidine base to the uridine base. So the cytosine is converted into uracil. So here the endonuclease function of the Cas9 is modified, and it is fused to a cytidine deaminase. The modified Cas9 is either a deactivated Cas9, okay, or a nickase Cas9, which has its endonuclease property deactivated, means it can't cut anymore, and it has the deaminase which can convert cytosine to uridine, which results in the C to T transition in the genome. So the steps involved in the CRISPR-Cas9 based genome editing. So the first of all, if suppose one want to perform an experiment in genome editing, or want to do a genome editing to improve a particular trait, so for that a gene has to be selected, and the guide RNA has to be designed. And for selecting the gene, mostly the negative regulators they are targeted, and these are the different software which are used for the designing of the sgRNA, like CRISPR-P, CRISPR-Plant, eCRISP, ChopChop. Okay, these softwares will give you the guide RNA sequence. Then we synthesize this guide RNA, and then we clone it. So this guide RNA is synthesized and cloned in a Cas9 vector. So then we do the single and multiplex gene target. So we can target a single gene also, and we can target more than one gene. So that is multiplex. So after these steps, the The construct is delivered into the plant. That is delivery method for the host system, and we can use the stable genetic transformation also, and we can use the transient gene expression also. So we can use the agrobacterium mediated transformation or biolistic method we can use, or we can use the floral dip in case of Arabidopsis. And or in case of the transient gene expression, we can do the micro injection or protoplast mediated method. So when the this construct is delivered into the host system, then we screen for the transformants, and then we screen for the mutations. To screen for the mutation, we can use the restriction enzyme based assay, we can use the Sanger sequencing, or we can use the real-time PCR also. Then we can evaluate for the target trait. Suppose the target is the abiotic stress tolerance, then we can do the phenotypic evaluation. So these are the steps for the genome editing experiment. So these are the different vectors which have been used, and there are the P green based vectors and the P Cambia based vectors which has been used for the CRISPR experiments. So this is how we perform the experiment. This is the sequence. Suppose this is the viral sequence. Then we select the guide RNA using the PAM sequence, that is NGG. Then after selecting the sequence, we clone this sgRNA in the vector. We clone them. Then we confirm that whether the sgRNA is cloned or not by the restriction digestion and PCR. Then we subclone them in the binary vector. So and after the subcloning also, the construct they are confirmed using PCR and restriction digestion. And this is the work which we are doing in tomato, that is targeting of the lycopene cyclase genes. So we have taken these three genes, lycopene cyclase beta 1, beta 2, and epsilon. So we have designed these sgRNA sequences. So these sgRNA sequences have been designed, and then we have cloned these sgRNAs in the sequencing vector. And after this confirmation of this cloning in the sequencing vector, they are cloned in the p-direct 22C vector, which is the Cas9 vector. And after the confirmation of the cloning in the Cas9 vector, this construct was used for the transformation. And this is the Pusa ruby. So in this genotype, the construct was used for the transformation. And these are the transformants which were obtained on the selection media. So these transformants were checked by the PCR and by sequencing, and these are the Cas9 transformed plants obtained. So regarding the regulatory part of the genome editing plants, so they are in some countries, they are not under the in the genetically modified organisms, like US and Japan. They are not considered there as genetically modified organisms, but they are under strict GMO regulation in Europe. So the difference between the genome editing plants and GMOs is that the genome of the edited plants can be free of the exogenous DNA sequences. So we can eliminate the sequences from the genome editing plants, and the mutation created is as good as the mutation created by any other method, like X-ray mediated method or any other method. So so we can eliminate the foreign DNA sequences. So the exogenous DNA of the editing tools can be removed through the genetic segregation. or may never have to be introduced. So if you are introducing the CRISPR reagents as ribonucleoproteins, so the foreign DNA need not to be incorporated. So therefore they are different from the GMOs. So in case of the some crops which are clonally propagated, like potato, sweet potato, and banana, so the elimination of foreign DNA fragment is sometimes difficult, but one possibility is that we can generate the transformants by the high throughput screening. That we have to screen the more number of the transformants, or we can use the other delivery methods, like we can use the in vitro transcript or ribonucleoproteins in these crops. In case of horticultural crops, in 2013, the first example of the genome editing was achieved, and the tomato has received much more attention compared to other crops, and compared to other crops. So most of the studies they are from the tomato, like 42% and 13% from the potato, and among all the horticultural crops, the vegetables, they constitute 70% of the genome editing studies, while the some floral and medicinal plants, they have also been manipulated by the genome editing. So these are the different traits which were manipulated using the genome editing. So these are the traits related to the plant growth and development, and in these crops, and these are the traits which are related to flowering and fruit quality. Then these are the traits which are related to the biotic and abiotic stress tolerance. So these are the traits which are targeted in the horticultural crops. So these are the other examples. So the mutants which we generate through the genome editing, they can be directly used for crop improvement as pre-breeding materials, and and the desirable traits, they can be directly introgressed into the elite lines without compromising other properties. So the resulting lines from the genome editing, they can be used for the targeted improvement. They will be ready for use in production. Also, the wild relatives of the cultivated varieties, they are also the potential materials for the genome editing. For example, in case of the tomato, the Solanum pipinifolium, it was domesticated by the editing of the several important genes affecting the plant architecture and fruit development, resulting in the new tomato varieties, which has the desirable properties of the cultivated tomato combined with the favorable traits of the wild species. So these are the other applications, like mutations can be directly introduced into the coding regions or the promoter region, that is the cis regulatory regions, and which can be used to generate the variations for the breeding selections. And in case of the tomato, the fruit locule number is determined by the several naturally occurring mutations in the cis regulatory region of the clavata Wuschel. So in the Wuschel promoter, the locule number is determination of the locule number is located. So this finding motivated researchers to design a multiplexed CRISPR-Cas9 system targeting the clavata Wuschel promoter to generate tomato lines with a wide range of the locule numbers. And these variations has also been observed when the genes responsible for inflorescence and plant architecture are engineered. Along with the Cas9, we can target the epigenetic region also. The epigenetic status of the region can also be modified. So when an sgRNA is fused with the dead Cas9, and the catalytic domain of this translocation demethylase, it can remove 5-methyl cytosine at the specific sites, increasing gene expression. So epigenetic mutations can be created using the dead Cas9, and then the epigenetic recombinant inbred lines can be created. So, so the individuals from these populations, they are genetically identical, but they are epigenetically distinct. So these epirils, they have been constructed in Arabidopsis, and they exhibit considerable phenotypic variations. These examples, they showed that genome editing is an excellent tool for producing new alleles as well as epialleles, where the epigenetic modifications are done, and which are important sources for phenotypic variation for crop improvement. Okay. Then there are RNA editing factors which are involved in C2U editing. So these are the different RNA editing factors like PPR proteins, or multiple organelle RNA editing factors, or organelle zinc fingers. So these are the different RNA editing factors which can do C2U editing. Like base editing, here the RNA editing factors can be targeted. So, so this is an example where this loss of the SLO RRM4 mutants using the CRISPR-Cas9 gene editing systems. So this different kinds of the phenotype in fruit can be created. So this is the wild type, and this is the mutant which is showing the delayed ripening. So this is the network of the gene editing method, that what we can do, that we can do the gene knockout, we can do the gene insertion, we can do the gene replacement. So these are the different gene editing methods like ZFN, CFF1, Cas9, base editor, TALENs. So we can do multiplex editing, we can do high throughput mutant libraries, we can do the gene regulation. So this is the overall strategy one has to follow. We have to design, then we have to make the construct, then we need to deliver the construct to the plant, then selection is required, whether the transformation has been done or not, and then the edited plant. Coming to the regulatory framework, that what is the regulatory framework for genome edited plants in India? So the genome edited plants which are derived from use of the genome editing techniques like ZFN, TALENs, CRISPR, and other nucleases, they have been classified under 3 categories. So first is STN1, that is site-directed nuclease 1. So here it is site-directed mutagenesis without using a DNA sequence template. So here a DNA sequence template is not used, and without template the mutation is created. In STN2, site-directed mutagenesis using a DNA sequence template, and in STN3, site-directed insertion of a gene or large DNA sequence using a DNA sequence template. So in STN2, mutagenesis is created using a DNA sequence template, and in STN3, insertion or deletion of the large DNA fragment is done using a DNA sequence template. So, so these are the 3 different kind of the categories. So now coming to the STN1. So the STN1 involves the unguided repair of the targeted DNA break by the natural endogenous DNA repair mechanism of the host, such as non-homologous end joining. So this spontaneous repair of this break can lead to a mutations which can cause gene silencing, gene knockout, or change in the activity of a gene. So the STN1 genome edited plants will be free from exogenous foreign DNA, and these mutations can be base substitutions, indels, deletions, including large deletions or structural changes. So the resultant mutants, they are comparable to those occurring in nature. So this is the important point here, that the mutations which are created using the STN1, they are comparable to the mutations occurring in nature, or mutations obtained through the conventional mutagenic treatments or natural variations which are found in the primary or secondary pool. So these mutations are comparable to the mutations which are occur in the nature, or which occurs through the conventional mutagenic treatments or natural variations. So in an STN2, it involves a template-guided repair of the targeted DNA break using an externally supplied DNA sequence. So here we provide a template to repair the DNA. So the donor template carries one or several small mutations flanked by the two sequences which match both the ends of the DNA break. So it recognizes a repair template, allow the introduction of the mutation at the target site, and the resultant mutant carries modified sequence leading to an altered expression profile of the gene or the altered activity of the encoded protein or RNA. Thus, the edited version could be regarded as the allelic form comparable to those available in the primary and secondary gene pool. Okay, so here also the edited version is can be regarded as allelic forms comparable to those available as primary or secondary gene pools. So in STN3, it involves a template-guided repair of the target DNA using a sequence template, typically double-stranded DNA containing an entire gene. Here you have entire gene or even the longer genetic element. Both ends of the template sequence are homologous to the DNA breaks, which allow introduction of the target sequence of the gene or genetic element at the target site. So the STN3 genome edited plants, they have foreign genes in a specific locations of the genome conferring new traits. These products will be clearly distinguished from the naturally existing variations or mutations in the species. So STN3 generated plants, they are different from the naturally existing variations or mutations in the species. So these STN3, they will be under the GMO regulations, and STN1 and STN2, they will be free from the GMO regulations.
Ma'am, time is up. Kindly please void of it.
Okay, okay. Thank you so much. I have completed. If you have any questions, then we can take.
Any questions, please ask.
Thank you, madam. Wonderful presentation. Nice information.
Okay, thank you so much.
Good morning, ma'am.
Hello.
Good morning.
What is the difference between gene editing and gene modification, ma'am?
Using genome editing, we can do the genome modifications. So in genome modification, we can talk about modifications. So these are the modifications that either we are creating any insertion, or we are doing the deletions, or we are replacing the entire fragment if we are doing the gene replacement. So these kind of modifications, they can be done through the genome editing systems like CRISPR-Cas9, CRISPR-CPF1, or base editing, or previously ZFN and TALENs.
Thank you.
Okay.
Hello, ma'am.
Yes.
Hello. Can you
Yes.
hear me?
Yeah.
Yeah.
Yeah. Yeah. Yeah. I can hear
Ma'am,
you.
thank you for this wonderful presentation. I'm Dr. Ishtiaq from Kashmir, KVK Anantnag. I'd like to know, is there any major horticulture crops, including temperate fruit crops, which have been, manipulated through this Cas9 so far successfully, and for what traits? And how
Uh,
do you see, gene editing as a tool for breeding or manipulating these traits in future, in particularly in our country so far? How success we have achieved when it comes to gene editing through Cas9? Thank you.
okay. Thank you for this wonderful question. So to carry out a genome editing, first of all, we need a target gene. We should have a target gene, and that sequence of a target gene should be well characterized. Okay, so suppose if you want to target for fruit quality, if you want to improve fruit quality, or you want to target for pest resistance, or you want to improve any abiotic stress tolerance, so for those traits, the target genes, they need to be well characterized. After uh, after you have the target genes with you, you should have the transformation protocol of the crop in which you want to work. Suppose if you want to work in apple, you should have the well standardized transformation protocol and regeneration protocol. So if you have these two things, then you can go for the genome editing. Okay, so you should have the genes which you want to target, then you should have the transformation protocol and regeneration protocol, then you can go for the gene targeting experiments.
Any major fruit crop, as I told you, which has been manipulated through this in
uh,
India,
yeah.
including some temperate fruit crops?
Yeah, like there are you can actually we work in vegetable crops, but there are examples you can search in apple. In apple, there are examples. You can search for those examples, and in citrus fruits, there are examples. Papaya, there are examples. But in case of, if we talk about horticulture, then the major work has been done in case of tomato, because in tomato the transformation system is very well characterized. So the most of the work has been done in tomato and potato, but obviously there are crops, there are fruit crops which has been targeted using this system. And now the more and more studies are coming, as we have discussed that this is a simple technique. If in any lab if we can do cloning also, then we can do this genome editing also. This as simple as that.
Any other question? Thank you, ma'am. Thank you on behalf of the dignities. Thank you on behalf of NADCL. Thank you on behalf of the students, and thank you on behalf of the organization community. Thank you so much. We will conclude this session here. The link of for the second meeting has been sent to your WhatsApp group. Kindly check. Please join there. Second
Okay.
session will start now.
Okay. Thank you so much for providing the opportunity. Thank you.
Thank you, madam. Thank you.
Thank you.
Hello.